This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 91, recorded on November 3rd, 2014. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hey, hi, how you doing?
Haven't seen
All
you.
is well there?
Yeah, everything's well. I haven't heard you in a while. You've been traveling.
I know, I've been traveling and keeping busy. Yeah,
It's always
but
good.
I'm back.
I was out your way last weekend.
You were in San Diego?
I was in La Jolla.
In La Jolla, no less. What were you doing?
I was at Scasm.
Oh, that's, of course. I was supposed to be there. Scasm being?
Southern California branch of the American Society for Microbiology.
You taped a show there, didn't you?
Yeah, we did a show with two really interesting people.
Yeah, I heard some of it. Yeah.
And I had a great time. I met a lot of clinical microbiologists who are very nice people. They're very enthusiastic. And I must say the weather was gorgeous out there.
Tends to be.
Here, it's cold and fall is here. All the leaves are going. Oh, well. Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How have you been?
Terrific.
Is the golf game well?
It's getting dark pretty early, so
Can't go, huh?
I think I'm going to have to put the clubs away for the season.
Oh, what a pity.
Really? It's too cold in the winter, I guess, huh?
Come see us in San Diego.
They don't make indoor golf places, right?
You can go to driving ranges indoors.
Oh, that's right.
But we also have an airport nearby.
Mm-hmm. Why would I think otherwise? You know, you have to go to Detroit to fly anywhere, right?
It's 30 minutes.
Yeah.
Door to door. I love it.
And you don't actually have an Ann Arbor airport, right?
There is a small airport. Yeah, Willow Run.
Mm-hmm. But you don't use that, right?
No, because, 30 minutes to Detroit Metro, and then we can fly nonstop almost anywhere. It's a hub for Delta.
And then you can go play golf where the weather's good, right? That's the whole
right.
idea.
Or go see my science friends.
Yeah. Well, you get around, right?
I do.
Do you like traveling?
I enjoy traveling. It's my children are independent now, so it's I can travel much more easily.
Yes, I look forward to that day. Almost a couple more years. Do they have to be out of college to be independent? I guess so, right?
Well,
These days, they have to be after graduate school.
I was talking to a student in our local high school, and she said, I want to major in neuroscience. What can I do when I graduate college? I said, you can go to graduate school. Otherwise, you're not going to be able to do much. That you could be a laboratory technician, but you're not going to want to do that your whole career, probably. Although some people
it's a
do.
Great option, though, right out of undergrad.
Yeah.
To get a taste of it.
Well, I told her she should decide if she likes it. She should work in a lab for a while.
Absolutely.
Right? Today we have, as usual, a paper, but we're going to start with something different, and Elio's going to tell us about it.
All right. So here's the idea. Microbiology is full of important facts and important work being done. And we focus on that. We certainly emphasize in this podcast the things that are important and so forth. But there is a class of phenomena which I would certainly not say are unimportant at all, but they are characterized by something a little bit different, namely being almost totally unbelievable, unexpected, surprising, and sometimes not well known. And we specialize in that more or less in our blog. So we thought maybe we would introduce every so often this podcast with one of these and do a snippet of it. Not go into great depth, but just sort of talk about it in terms of why the world of microbes is so gee whiz. I should say that J.B.S. Haldane, the famous scientist, said, nature is not only unpredictable, It is more unpredictable than we can think, something like that. So can I start this out with a snippet of the fact that off the coast of Chile, for the length of 3,000 kilometers, because Chile is skinny and long, there's a continental shelf about 100 miles wide. And on that shelf, if you go down to the depth, which is about between 80 meters and 300 meters, something like that. It varies a little bit, but it's fairly shallow as depths of oceans go. You find a mat of a hairy something. You see filaments sticking out as if they were a mat. And that is surprising. And for some time it's been known that this is made up of bacteria. Now, these bacteria, which are called thioploca, which means sulfur braid, I'll come to that, why it's called sulfur braid, make up an area and a volume and a weight which is almost unintelligible.
Mm-hmm.
It makes up 100 million tons of material, okay, which makes it probably one of the biggest biomasses of single bacteria. It's not just, well, there's a few kinds of bacteria there, but there's one major one usually. It's probably the single biomass of its sort, and certainly the biggest biofilm that anybody can imagine. And there it is, down there, quietly carrying out its own business. So what's this all about? And why are bacteria suddenly visible with the naked eye? It's supposed to be small, right? Well, what happens is the following. These bacteria grow in chains. They're themselves very large. They are between 10 micron in diameter, which is large for bacteria, to 40 microns in diameter, which is very large. And they tend to be fairly long as well. And they make chains of these filaments, which are centimeters long. And they are encased in a sheath, a polysaccharide gummy layer, which encases not just one of these filaments, but as many as 100. So you have something which is a collection of filaments within a casing, the casing being the sheath. And since 100 times 40 micron is very large, you can see these filaments with the naked eye. And they're very long. They are as long as 15 centimeters. Now, can you imagine diving down there and seeing this, wondering what on earth is going on here?
It probably looks like a plant.
It probably looks like a plant. Yeah, it looks like a garden of filaments. And it took a while, and there was a Chilean scientist by the name of Gallardo who studied this, and also it's been studied quite extensively. And the story is that these bacteria, let me talk about the bacteria Thioploca and their ilk. They are, by the way, cousins to the giant bacterium called Thiomargarita namibiensis. I don't think we ever talked about this on this podcast, have we?
No.
Okay, we should talk about this some other time. Anyhow, they're cousins, both gigantic. Thiomargarita gets to be 0.5 millimeter across, so easily seen with the naked eye. Anyhow, so much for bacteria being invisible, right?
Mm-hmm.
This only says, for every rule, there's an exception. Anyhow, Thioplocas, their business is to burn hydrogen sulfide. That's what they use for a fuel. They eat that. And they use as an electron acceptor. If you oxidize something, you got to reduce something else, right? So the reductant here is nitrate, not oxygen. Mind you, for many bacteria, nitrate looks like oxygen, or not quite that. At least it can act in lieu of oxygen and accept electrons, becoming reduced in the process to nitrites and so forth, eventually ammonia. Now, here's the story. You need two things then, okay? H2S, the smelly stuff of rotten eggs, and you need nitrate. Now, nitrate is found in seawater. It's not abundant, but it's found in seawater. H2S is not. However, down at the bottom of the sea, where there's a lot of biological material, organic material, which is being decomposed. That's an abundant source of H2S. Plus, there is some made geologically that seeps through. So the H2S is down at the bottom. The nitrate is in the seawater. How do you get the two together? Well, what happens is these bacteria have a rare skill. They move up and down their sheath. Okay? They commute between the bottom, which is H2S rich, and the top, which is nitrate rich. They go to work by this commute. This is probably driven by chemotaxis. You can show that they are chemotactic towards nitrate. And this is really sort of an unbelievable story. And it is huge, immense, gigantic, colossal, gargantuan. Want any more adjectives?
And I'm sure for Vincent, it brings the Lincoln Tunnel to mind.
Yeah, of course.
Very good. That's a very good analogy. If the cars on Lincoln Tunnel were all chained together, right?
Is this the only place on the globe where such a mat exists?
Well, at these proportions, yes. This is the biggest known, but there are mats like it elsewhere. Various other parts of the world, I find them. So you may want to look at this. Our readers would do well to Google Thioploca, 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 and you can find illustrations of this. It looks like when some of the pictures of these sheaths look like they are worms. You know, and by the way, I didn't quite explain them. They are stuck in the mud in the bottom in the sediment, the benthos as it's known, and so they stick out. Like have you seen pictures of marine snakes that go tail first into the sediment and wiggle around? Well, it's something like that. Only it's smaller, of course, but still. So anyhow, this is a snippet number one.
So people work on these mats, I presume? They study them?
They do. There's a fair amount known about their ecology. Their metabolism has been studied in quite some detail. Some things are still missing. I've been trying to see what is the composition of the sheaths. And people say they're polysaccharides, and that's about all they say. Gelatinous polysaccharides.
And they're mostly monoclonal or polymicrobial?
Well, they are the one kind of Thioploca tends to dominate. There are several species. One tends to dominate. However, there are other bacteria there. There are even protists that stick in and hitch a ride or whatever they do inside the sheaths. So there's some other bacteria which are apparently symbionts. It's not really known. And maybe even higher organisms. So it's not as simple as just saying it's a single culture. It's not a pure culture of the Thioploca, but it's totally dominates.
Yeah, totally. The photographs that you have looks very uniform.
Yeah. Over vast areas, they're uniform. But as I say, if you look within the sheath, you may find other things as well. It's an incredible. It's unbelievable in every sense. The ecological aspect of it, the physiological aspect, the structural aspects, wherever you look at Thioploca.
The scale is really impressive. Do they know at what rate this is expanding, or is it fairly stable, the size?
I don't know the rate, but I know that they move at a reasonable rate. Let me see. They move within the sheath. The organism move at something like a centimeter an hour, is it? Let me see. Hold on a second. Yeah, about 1 centimeter an hour. They can travel, which is, for bacteria, it's quite a speed. So, it's reasonable. So they take an elevator to work. They go up and down.
Does anyone try to eat this, like fish or something?
The problem, I don't know. The fishermen have known about this for a long, long time. This is not new. They probably fish them up when they they call them. There's a Spanish word for them called esparta, which means a dirty mat of something like that. It's burlap or something like that. It's or unwashed wool. So the fishermen had known about it, but the scientists took them a while.
That's cool. Very good. Thank you. I like that. So you have lots of these amazing, unexpected
we
things.
have plenty to go on. Believe me. we'll have snippets, all you want.
And maybe one time you can tell us about the biggest fungus in the world.
Oh, sure.
Or the tastiest.
Tastiest, too.
Okay.
Oh, that's what John Coffin said. Elio likes mushrooms. But he said when you were in Massachusetts, though, you had much more opportunities to get mushrooms than you do now, right?
Well, I tell you, mushroom hunting in Southern California is for masochists.
That's good. All right. Thank you, Elio. Very nice. I like that idea because for us, it's really curious to hear all this stuff. I'm sure our listeners will like it. All right. We have a paper today. It was published recently in MBio, and it's called Detection of Zoonotic Pathogens and Characterization of Novel Viruses Carried by Commensal Rattus norvegicus in New York City. And the authors are Firth, Batt, Williams, Fry, Simmons, Conte, Garcia, Buva, Lee, Che, Kwan, and Lipkin. And this is from my colleague Ian Lipkin here at Columbia at the School of Public Health. It's a collaboration with the University of Edinburgh. And it's an interesting story that plays out right here in New York City. I think most of our listeners will realize that a good number of human pathogens, whether they be bacterial or viral, come from animals. We call those zoonotic pathogens. And as as human populations grow and we get closer and closer to animals, we reduce the distance. We tend to pick up infections. In fact, when people first began living in bigger numbers many years ago because of agriculture and the ability to support larger populations, one of the things they did was to start domesticating animals, and they began to acquire their infections. So I would say now that pretty much all the viruses that we have as humans came from animals. So we need to know what are the potential future pathogens out there. So a lot of people are characterizing various animal populations. And in this particular paper, as you can tell by the title, they're looking at rats, the Norwegian or the Norway rat, Rattus norvegicus. And these
weren't rats, weren't they the guys who brought us plague? Michele knows all about it, by the way.
Yeah, they were. These animals in New York are called peridomestic. They live in and around human habitations, and they're very successful here in New York City. They live close to humans. They live in our buildings here in the city. I see them all the time on the subway, and Michele, you may remember.
Sure, if you look down in the tracks.
Yeah, they're always walking around, and they disappear into little holes. I always love to watch for them. You see rats, you see mice, but they stay down there. They can't really get up and come at us. So there are lots of them here in New York. I don't even know what the population of rats is, but I'm sure there are millions. They eat garbage. They reproduce to high numbers. We know that they have pathogens in them already.
Do they vote?
They don't vote. If they did, they would control the outcome. I'll bet they outnumber humans, right? There are 8 million humans here in New York. Maybe there are many more rats. I should look it up. So hantaviruses are known to be in rats. Bartonella, Leptospire Interrogans. That's an interesting one. You guys know something about that? Why is it interrogans?
I don't know.
Well, it's one of many leptospirals.
Interrogans.
They are also transmitted by dog urine. They're generally a tropical sort of a thing, but you find them in the temperate zone, all right. It's an interesting diseases. We should talk about it. We should bring somebody on, like Joe Minetz. He's here at UCSD, and he goes to Peru to study leptospirus.
Okay. Yeah, we could get him on. That'd be great. So the paper basically examines the diversity, the microbial diversity in rats. Because although we know there are some microbes that are in rats and humans can acquire them from rats, we don't know really how many are there and whether they pose a risk or not. So that was the basis for this project. And I know Ian has been interested in this for years. and he finally got to do it. So they trap rats in New York City. They trapped 133 rats from 5 different places in New York City. And as he says in the paper, they were places of convenience. So I assume this is where people lived in his lab.
Yeah, I missed that. They didn't go into what sites they.
Yeah, it's in the materials.
Yeah, it doesn't say.
It said these are all indoor sites. 3 of them were high-density housing complexes.
Oh, yeah.
One of them was a large indoor mixed-use space, and 1 small urban park. And he says in all these sites, the average, the median income is below average. And what they did was they trapped the rats, and then, of course, they sacrificed them. They took fecal pellets, serum, a variety of organ samples, and also they did some oral swabs so they could just see where these microbes were, whether they're respiratory or fecal oral, etc. So they first did polymerase chain reaction to screen for known pathogens. So you have to have a sequence, for the pathogen, the bacteria, the virus, and they use this to screen these samples. So they looked for 18 bacterial and 2 protozoan human pathogens. They did not find Campylobacter coli, Listeria monocytogenes. I'm sorry about that, Michele. Rickettsia, Toxoplasma gondii, Vibrio vulnificus, Yersinia pestis. Although these have been found in rats in other studies, they didn't find them here in New York City. And they found other pathogens each in at least 1 animal. 3 bacterial pathogens were found in more than 15% of the animals. So we have atypical enteropathogenic E. coli was found in 38% of the rats. Bartonella was found in 1/4 of the rats. And Streptobacillus moniliformis was found in 17% of the rats. And just a few words on these. Bartonella is, according to the paper, infects rat populations globally, and it is associated. Oh. You can look and find them in rats anyway. It's associated with human disease, including endocarditis, febrile illness, but it's not known what the role of rodents are in transmission of these infections. But here in New York City, in Harlem, a study was done of injection drug users. 47% of them were seropositive for Bartonella. And most of these infections are asymptomatic, but they say, well, maybe these bacteria are in rats. Maybe it's puts certain people at risk, intravenous drug users, immunosuppressed people, and so forth. So you will see throughout this paper, there's a lot of call for more studies because we don't know what the results mean. Leptospire interrogans was also found. You can look at this. You can actually find. This is an open access paper, so you can find it online. They found Leptospire interrogans in 16 rats. That's 12% of their 133. It was found in the bladder, the kidney, and in the urine. So you can imagine that is, as Elio said, in dogs it's spread by urine, and also apparently has the potential to do so in rodents. This is a highly prevalent and widely distributed zoonotic pathogen. Rodents are known to be the most significant reservoir, and humans get infection by consuming food that's contaminated with the bacteria, also water contaminated with rodent urine. You can also get it from bites of rodents or getting contaminated soil on your skin. So
I should say that if somebody goes to this table, they should not treat the incidence of the presence of all these bacteria equally. Some are more likely, like Leptospire is probably the best example of something that could be dangerous because it's not usually associated with the human body. But some of the other things like Clostridium perfringens is a very common bacterium, and it's not likely that if we come in contact with a rat-borne C. perfringens, we're in any danger.
Right.
So not everything, because it's a potential, most of these things are, some of these things are potential pathogens rather than actual outright pathogens.
Yeah. Well, they also make the point that some of these are known human pathogens like C. diff and Salmonella enterica, and they're probably in rats because rats eat human garbage,
right?
Including
right.
food.
So it's actually human to animal transmission is most likely.
It's an anthroponosis.
That's right.
An anthroponosis.
How's that instead of a zoonosis?
Yeah, that's a good. That's a
you
good.
You heard
name.
it here.
Because I've always heard it called reverse zoonosis, but I like your word better. Anthroponosis.
Well, and also in the case of the gastrointestinal pathogens that they only found in the fecal pellets, they may have just gone for the ride, but not be colonizing the rats.
Absolutely. Yes.
So it's not clear that it's a reservoir in those cases.
So the Salmonella was found in the fecal pellets as the
Shigella, Clostridium.
Right, which is where you'd expect if they're eating food, right? Yeah,
and the EPEC, to the extraintestinal pathogenic E. coli, was also just in the fecal pellets.
Right. Yes, so, this may have... The significance of all of this is really not known, but it's good to know what's there. Okay, they only... By this directed kind of sampling, which is polymerase chain reaction. We'll talk about another kind of approach in a bit. They only found one virus, Sol virus. It's a Hantavirus that is known to be in rats, and it causes a human disease which consists of hemorrhagic fever with renal syndrome. And this, here we are, it's on this table here, SEOV, and it was found in the brain, kidney, liver, lung, oral swab, salivary gland, serum, spleen, fecal pellets. So everywhere, it was not in the bladder or in urine, and it was not in the lungs. They sequenced this viral genome, and the sequence reveals that it's highly related to a strain that circulates in the United Kingdom and probably came over the pond 3 to 16 years ago. And both the UK and the American strains originated in China. So how it got here, you can guess, probably rats getting on a ship, I would suppose, or people bringing it over, who knows. Anyway, this is the first time this virus, Sol virus, has been identified in New York City. Okay, next they did deep sequencing. So they take serum, fecal pellets, or rectal swabs, and they keep these two things separate. And they make pools of 4 to 6 samples, and then they deep sequence and ask what viral sequences are here. And they focused on the mammalian viruses because they also find phages and insect viruses, plant viruses, fungal viruses, whatever the rats eat has viruses in it, and the rats will have them, but they didn't pursue any of that. And Table 2 is full of all of these viruses, many, many different viruses detected in this study. In the serum, they found 4 families of viruses. Now remember, these are all sequences. Nowhere in this paper do they actually isolate virus. So if you were really curious as to whether any of these exist in rats, you would have to either look for antibodies in the serum or actually isolate the virus. Anyway, in the serum, they find 4 families. Anelloviridae, these are small single-stranded DNA viruses that virtually all humans are infected with, and apparently rats. Boca viruses, recently discovered agents of respiratory disease in people. Adenoviruses and parvoviruses. These are all common viruses. It's not surprising to find them there.
Let me ask you, isn't it funny there are not more DNA viruses, given that there's such a great propensity to go into latency and be carried around and all that?
Yeah, I don't know why they're certainly the preponderant virus in this study, as we'll see in a moment, are RNA viruses. Yeah, so I don't know why, but I mean they have some. These viruses are all DNA viruses that we mentioned. So
the DNA viruses?
Yeah, Anello, Boca, Adeno, and Parvo are all DNA viruses. Yeah. Okay. In the fecal samples, they find Boca viruses, Calhi viruses, cardioviruses, Circos, which are also small DNA, honey virus, HUNNI, astroviruses, adenoviruses, parvoviruses, and rotaviruses. And they say many of these were similar to known rat viruses, so they didn't bother with them further. However, a lot of the sequences had less than 70% similarity to known viruses, and they pursued those and tried to learn more about it. In particular, there were a number of viruses which belong to the flavivirus family. This is a family that contains, for example, yellow fever virus. And in particular, hepatitis C virus is a member of a genus in this family called the Hepativiruses. They found 2 novel Hepativiruses and a couple of other novel flaviviruses in 36 of the rats. Now, this is an interesting observation. 19 of their rats had just 1 virus. 10 rats had 2 viruses, and 6 rats had 3 or more viruses. So multiple infections prevail, which shouldn't be surprising because we know, at least in humans, we have lots and lots of viruses in us. And again, this is all detected by sequencing, so we don't know if there's in fact infectious virus there. The Hepativiruses are distantly related, that the ones that they found 2 novel Hepativiruses in rats. They're distantly related to hepatitis C virus, which of course is an important human pathogen. And they looked further and they found actually evidence for viral RNA in the liver. And in humans, of course, hepatitis C virus is a hepatotropic virus infection. And there isn't really a very good animal model for hepatitis C virus. A humanized mouse model has recently been developed where you put human immune systems into mice. But there are some problems with that model. And so they're wondering whether this rat would be a good model for infection. Now, of course,
scientists to investigate the disease, the mechanisms. That's exciting.
So they would have to actually recover the virus and then show that it infects rats and gives them hepatitis and so forth. And if all that worked out, this would be kind of an interesting model. Now, the origin of hepatitis C virus is a bit obscure, and in recent years, people have identified equine and canine relatives of hepatitis C virus. But interestingly, those viruses don't replicate in the liver of horses or dogs. So this rat Hepativirus, does appear to be in the liver. So this could be quite interesting.
How about retroviruses? I thought our genomes are loaded with endogenous retroviruses.
It's interesting that you should ask that, because I had a big conversation with John Coffin, who is very interested in that sort of thing. They didn't find any retrovirus sequences in any of these tissues. Now, I'm sure rats have endogenous retroviruses in their genome, but that's not what they looked for here. By looking in serum and tissues and so forth. So no mention of retroviruses, but I'm sure
I think if I'm reading the materials and methods carefully enough, it looks like they took the serum samples, fecal pellets, rectal swab samples, and then first enriched for viral particles before they did the sequencing.
Yeah,
so it may be that they just did not look in the rat cells to find the retroviruses.
So that would explain
must be it. Yeah. That's probably
they might have just not had the sensitivity.
Yeah, if they enriched for particles, for sure. But you would definitely expect to see endogenous retroviral sequences in the genome of rats, but no particles. So my guess, Elio, would be that like in humans, all the endogenous retroviruses are defective. They don't make infectious particles.
Yeah, but still, look, it says here, DNA and RNA were extracted from each tissue and fecal sample. So they did extract from tissues.
For which experiment?
It's in the materials and methods.
But was that what they did the whole genome sequencing of?
Targeted molecular analysis.
Yeah, that's the PCR.
Mm-hmm.
Yeah, but why don't they find them?
Well, the PCR, let's see, the PCR was done for 18 bacterial and 2 protozoan.
Depends on what primers you use, obviously. But
and then further down under what they call...
Why would they do retro?
under UHTS, the section, Materials and Methods section, which is where they did the sequencing. That's where they described, they first enriched for viral particles.
That's right.
I see.
So the sequencing they did enrich. And so
I'm fine with this.
That would get, otherwise, if they did total DNA, nucleic acids, well, that would be, you get a lot of noise. Yeah. But so you should find endogenous sequences for sure, but their methodology, I think, rules it out. It's interesting, though, similar story in bats, Elio. You find endogenous retrovirus sequences in bat genomes, but you don't find particles in the animals.
Well, that's true for humans. I mean, you don't find endogenous particles of endogenous retrovirus in humans, do you?
I asked John Coffin that, and he said sometimes you do. Sometimes,
Is that so?
Yeah, you can see them in the placenta, he said. And so in some tumors, you find they're not infectious, but they're particles, retrovirus particles. So apparently we can make particles, but they don't transmit. Yeah, maybe that's a snippet. All right. So they then studied some of these viruses that were distantly related to known ones, and as I said, the flavies, the hepatitis C was one of them. And they find lots of picornaviruses, which is very enheartening to me because this is the viruses that I study. So I'm happy to see that they're in rats. Of course, only because I like them, not for any other reason. They found many different picornaviruses in the fecal sample.
Not accounting for taste, by the way.
Yes, that's true. And these viruses, some of the picornas are transmitted fecal orally. Some are respiratory, but they found a lot in the feces. They found Kobu viruses, which is one of the genera of picornaviruses. And in particular, they found half of the rats had Kobu viruses, again, which are picornaviruses like polio. And one of them was related to a virus found in sewage in Kathmandu. How about that?
Huh.
We have rats from Kathmandu here in New York City. Not really. Some of the Kobu viruses are known to infect humans. They found a number of novel ones, new picornaviruses that hadn't been seen before. For example, one is called a parechovirus. These are in humans. They can cause respiratory and gastrointestinal infections. They found a brand new one in rats, and they call it the Manhattan parechovirus. Isn't that clever? Manhattan parechoviruses. And in mice, we know that lots of these picornas can be found there. So basically, 119 of 133 rats were positive for at least one microbial agent, with an average burden of 1.6 bacteria and 3.1 viruses per rat. Only 10 rats were infected with more than 2 bacterial species. 53 rats were positive for more than 2 viruses. 13 rats had more than 5 viruses. And they found as many as 9 different viruses and 4 different bacterial species in the same rat. And they had 1 rat with 11 different microbes in it. Just so you know, there are lots of microbes, bacteria, and viruses in rats. Some of them are known to cause disease, but for the rest, we don't know. And what they say in the paper is we should keep our eye on them. We should keep sampling, and we should check people to make sure they're not being infected.
Let me ask you a question. They didn't look at laboratory rats, or for that matter, pet rats. You can buy rats in pet shops.
Yeah, they didn't.
You know anything about them?
They didn't say anything.
Maybe along the same line, they took rats, which of course we all vilify, but what if they'd done this experiment with cats or with humans? Out of 133, how many would be positive for some virus?
Oh, all of them. Everybody has at least one virus.
Right.
We all have about a dozen viruses in us.
So I'm no fan of rats, but
sure.
I don't want the listeners to get the wrong impression that rats are unusually burdensome.
Dirty.
Right. No,
Yeah.
the point here is that they live among us, right?
Right.
So we need to know what's in them. I guess you could say the same about cats, right?
Right.
And cats can transmit infections
Or how
to humans.
about pigeons? New York City pigeons.
I think Lipkin did a pigeon study not too long ago.
Because
Yes.
pigeon poop is everywhere, on windowsills,
Yeah.
near air conditioning.
It's aerosolized, right?
Yeah, I'd be more worried about that myself.
Let me look very quickly here. I have a feeling if I do a PubMed for Lipkin and pigeon,
In New York City.
I will find something. I just remember him doing this study. But I personally, Michele, think that we should look in all animals out there to see what they're carrying, just out of interest. And I'm sure we would discover new things. But I do think we should be aware of what's around us. As I said earlier, we get closer and closer to animals. No, there are no papers if you search for Lipkin and pigeon. So he didn't do it. How about if I try one more search? Pigeon.
But to your point, I'm sure our listeners are following the Ebola outbreak, and I was surprised to learn that some of the patients here in the U.S., their dogs had been quarantined.
Right.
And I guess this was a precaution. They were concerned that the Ebola virus would spread from the human to the pet. And I understand that. And so in thinking about this data too, our tendency is to think that the rats are giving us these diseases, but we could have just as easily been the source of the viruses and the bacteria.
In fact, there's no evidence that any of these microbes are dangerous for humans.
Right.
And they point that out in the paper. This is just more about knowing what's in them. I found a paper where Eric Delwart over in San Francisco in PLOS One in September 2013 published the viruses of wild pigeon droppings. 51 urban pigeons from Hong Kong and Hungary. And they found tons of viruses in them. Well, both picornaviruses, as we've shown here, and a couple of DNA viruses. So, yes, Michele, there's a lot of viruses in pigeons, and in pigeon poop in particular.
Yeah, and pathogenic fungi.
Yeah, what is the name of that one in pigeon?
Some bacteria also, right? Aren't birds?
Yeah.
I think Cryptococcus.
Cryptococcus, maybe, yeah.
Cryptococcus.
Yep.
So, I mean, I think, Michele, wouldn't you agree you should stay away from rats, though, the wild rats?
Absolutely. And I think our public health inspections of restaurants to make sure there are no rat droppings in the food storage area is a wise investment.
That's one of the things they point out in this paper, that rats not only eat human waste, but they're also in areas of food preparation and distribution. So the opportunity for reintroduction is there. And we're obviously not going to get rid of them, but we need to just think about what could potentially happen. Anyway, I thought that was a cool paper. So now you know.
I guess historically the Black Plague, which many of our listeners may be familiar with, that the reservoir was rats, and it was an urban disease, but there was a vector then. There were fleas that fed on the rats, and then if they bit the human, that was the route of transmission.
Right.
So it wasn't rat bites or rat urine that spread the disease directly.
All right. We have a few email to read here. The first one is from Jacob. Hello, hosts of TWiM and TWiV. I'm sending this to both podcasts because I'm interested in hearing what all of you have to say. I saw this question on Reddit today, and I thought I would put it to you as well. Here's the question. If you could convince the entire human population of one thing, what would it be? Elio, what would you try and convince the human population of?
Well, practice birth control.
That would be hard to convince.
That'd be hard to convince. Go through the streets at night yelling, "Stop it! Stop it!"
I don't think you could convince everyone of one thing. That's the problem.
A hard one. But is the question meant to be microbiological?
It could be whatever you want. Since we are a microbiology podcast, you could make it microbiology for sure.
Well, there's a number of diseases that you could eliminate by altering human behavior. HIV being a good example. You could just get rid of it. Just if overnight people altered their behavior regarding HIV transmission, there'd be no HIV.
Yep. How about you, Michele?
I guess I would take a page from the paper that we just read and convince everybody that we're all connected. So certainly we've seen that with the Ebola outbreak, that what happens in Africa is a concern of ours. We're all affected, and what happens in the animal, how we treat animals, how we use antibiotics in animals affects humans. So kind of the interconnectedness.
That would be like One Health, right?
One Health, yes.
Yep.
Good point.
Which is a movement, I guess. How would we describe what One Health is? It's a
It is
philosophy.
It's
a
an approach.
great question. It's a really fabulous question.
It's an approach to health, to maintaining healthy people, right? And you do so through animals as well as people.
By monitoring what's happening in the animal populations.
I heard the best example of One Health was from Linfa Wang, who's a virologist in Australia. So they made a vaccine against Hantavirus. People get Hantavirus infection from racehorses who get them from bats. So they immunize the racehorse and the people don't get infected. And you have to immunize far fewer individuals that way. That's a true One Health solution, right? So the same with Ebola. If we could figure out how that first Ebola case arises, from a bat or bushmeat consumption, we could somehow interrupt that, not necessarily by immunization, but by some other means. And that would be a One Health solution. So that's a good one. I like that very much.
How about you, Vincent? What would you convince the entire human population of?
Get vaccinated. For the vaccines that we have, take them. They save lives. Influenza, measles, mumps, rubella, etc.
I got my flu vaccine here at the medical school last week.
Yeah, I did too. Especially cancer vaccines, hepatitis B virus, human papillomavirus. There's so many people who don't want to get vaccinated because they have heard stories, and most of these stories are not true. So that's what I would do, because it saves lives. I'm not sure.
It protects others.
It does.
Not just yourself. Yeah.
Herd immunity. All right, the next one is from Jenny, who writes, I've realized that with all of the learning that I do on digital media, that I don't always do the best job of writing notes as I used to in the margins of books, for example. This relates to your inspiring podcast, since it is a major means for me as an RN to understand our relationship with microbes. I know that at this point you are barely started with the process of TWiM transcription, but just for the sake of daydreaming, what would be a best-case scenario of your audio backed up by an interactive transcript? When I think about the best possible learning tools that grow and change with the learner, I would like to see two things. First, a generic important points at a glance that shows up whenever you take a peek at what you've heard. You have a start with that, but it's not complete. Second, I wonder if there's a way to have a reader, listener's notes on that piece, once again, that show up at a glance on a transcript or marked to listen to again on audio. Often the epiphanies of TWiM are hidden in offhand comments. Then I have to ask myself much later, dang, which TWiM was that in? By the way, I've been listening from the very beginning and listened to the whole trifecta regularly, TWiM, TWiV, and TWIP. My tech-savvy husband showed me the old-school client that he uses, Thunderbird, and we installed quick notes on this email client without the email. We were able to write a note on the Prochlorococcus, and the note was stable on completely deleting the RSS for TWiM and reinstating it. But I'm still at the whim of continuously aging, changing tech that can disappear, and my notes along with it. I want my notes to grow with me as I go through life as an adult learner and attached to the original document interactively. If anyone has any ideas on capturing the elusive audio epiphany as an enduring note, let me know. Thanks, Jenny.
How thoughtful.
This is a great question.
Really great.
And this is one of the major issues with a podcast. Anything written, let's say Small Things Considered, you do a Google search for a term, you're going to find it. You can't do that with podcasts because Google doesn't index them.
But there is Google searching now with images.
Right.
So we're getting closer.
So my friends in the podcast industry tell me that at one point there will be, Google will index the podcast and you'll be able to search it at some point. I don't know when that's going to happen, but obviously that would be ideal because right now all that content is not searchable. And what a shame, right? Because there's lots of good content. So you could search on one thing that Elio just said, in today's podcast and find it instantly. And I'm sure that that is doable. So a computer can zip through the digital audio file and just index all the words. And there you would have it. So that's what I'm waiting for, Jenny. In the meantime, you're right. Everything is kind of a hack and depends on too many random things.
And as a non-for-profit, we're unlikely to be at the forefront of that technology at ASM. Right?
No, but Google will do it.
Right.
That's what they're in the information business. And there's so many good podcasts now that we have to do that.
Yep. Once the tools are available, we'll hook up.
All right. Bradley asks, how long can food sit on a counter? The conventional wisdom is that Thanksgiving dinner leftovers must be refrigerated within 2 hours. And he gives a link to an ABC News story. True or false? I don't know. I put it away as soon as I can. What do you guys think?
I would say it's mostly false.
So 2 hours, it can go longer?
Yeah. I mean, most of the food is cooked.
Well, it depends on the food, or how it was cooked. Depends how long you keep it. But you put it in the refrigerator, you wait 2 weeks. It's not a good idea, no matter what.
Yeah.
So how long are you going to keep it in the refrigerator?
And are you going to have it covered or not open, exposed to the air where molds and things can drop onto it?
Did you use your regular cutting board, which is probably the most contaminated part of your house? It depends on so many things. You can't really come up with, I doubt you can come up with an absolute statement. You can say, do the best you can. I mean, don't leave it out more than you have to.
This ABC News article, the title is "Leftover Turkey to the Fridge Stat." So they mean right away.
But Elio, we should be able to give some general guidelines about foods that are the highest risk for food poisoning, like at a picnic. I mean, it's like potato salad, right, with mayonnaise and
eggs, right?
Yes, things that staff can grow in. I don't know.
I would say anything that's been sitting out at a picnic for hours, you probably should pitch.
Yes.
But, when I cook a turkey at Thanksgiving, it sits out in the dinner, I don't know, hours and hours. And then in the evening, I carve the rest and put it away. And it's usually fine for a week. So as everyone says, it really depends on the food, right? Broccoli is probably not going to be an issue.
Right.
Too bad.
Well, anyway, that's our advice, Bradley.
Good question.
Justin writes, has there been any work to show the changes in the microbiome when someone takes a multivitamin? And I do recognize that all multivitamins are not created equally. I want to use this knowledge to assist me in picking my supplements more intelligently. Thank you so much for everything you do on the podcast. If you just want to respond with an article, that would be fine. Well, Justin, I couldn't find any articles in PubMed, and I asked one of my colleagues here who works on the microbiome, and he said it hasn't been looked at.
Well, there's an article in the New York Times, just came out, I believe it was yesterday, that talked about this idea that this is an article by Ed Young, came out November 1st.
Yep.
There is no healthy microbiome. And he makes a fantastic point. There's no such thing as a definition of a healthy microbiome. So what do you know if you look at a bunch of people who take supplements, a bunch of people who don't? You may find a difference. You may not. It doesn't tell you anything.
Yeah.
It's not useful information at this point. It may be someday, but right now, there is no such thing as defining a healthy microbiome.
And the reason is that there's just tremendous variation from person to person.
Right.
So to be able to pick out a trend when some of those people are taking multivitamins would require just an enormous sample size, and we just have not done large enough studies to have the sensitivity to pick out if there would be a trend.
Right. I really recommend this article. November 1st, there is no healthy microbiome. New York Times, Ed Young, that's Y-O-N-G. It's really a wonderful example of how to think about the microbiome. There's just way too much hype about it.
I find this really interesting because, as both of you surely know, hospitals everywhere are starting personalized medicine programs. And
right.
a big part of it is based on your microbiome. And I just don't see how that's going to work. I mean, I can see looking
but it
at...
may someday, there is enough sequencing power and computer power, you may be able to come up with some rules, but it's a ways off. It's not there yet, and it's not going to be there.
The challenge, Justin, is that right now most of the data are based on just DNA sequences, who's there. But what we really need to know is what the bacteria that are there are doing, what metabolic pathways are operating. And so it's less important that we know what species it is and more important to know what metabolic pathways are firing, probably. And so that's going to take yet another generation of experiments. So we're on it. It's an exciting idea, but it's premature. We don't know yet.
So you can just pick your multivitamins, Justin.
Yeah.
Doesn't matter. All right, last one is from Jesse, who writes, my department recently invited Dr. Susan Erdman from MIT to speak, and I thought she would make a very interesting guest for TWiM. She spent a good chunk of her day chatting with graduate students here, including stuff about how gut microbes might influence food cravings, which reminded me of your discussion in episode 86. Part of her research is on microbial effects on host hormones. Just a suggestion. Jesse is the author of Bacteriophiles, which is one of our sister podcasts on the Microbe World Network. Thank you, Jesse. Do either of you know Susan Erdman?
I don't know her personally.
I don't either.
You know, this, I don't know if you remember this paper we did. I think Michael Schmidt did it, where it was a review article, I suppose, where they were trying to explain cravings with the microbiome. And Elio was not impressed, right? If I should. Anyway.
Well, call me a skeptic.
Skeptical, that's fine. It'd be great to have Susan on. Sure, why not, Jesse? Sure. Thank you. All right, that will do it for episode 91 of TWiM. You can find it in all the previous 90 episodes at iTunes and also at microbeworld.org/TWiM. And we do love getting your questions and comments. Send them to twim@twiv.tv.
Let us know if you like the idea of these snippets that we did today. It's an experiment, so we'd like some feedback.
Yeah, do just send us an email. Say, I love the snippets.
Or I did.
Or I didn't like it. Yeah, I'd love to know. Let's see if our audience will respond. Michele Swanson is at the University of Michigan in Ann Arbor. Thank you, Michele.
Thank you.
Pleasure to have you. And Elio Schaechter is at the wonderful blog Small Things Considered. Thank you, Elio.
Thank you. A pleasure.
I'm Vincent Racaniello, and you can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and in particular Chris Kandayan and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.
So what did you think?
I think the snippet is great, and, we're always going to ask questions, so it's going to be 10, 15 minutes.
Where do you find these, Elio?
They're all in the blog.
I know, but you have to find them somewhere to put them on the blog, right?
Well, let me tell you, let me describe what I do. I sit at my desk and I look at the tables of contents of journals, and I look at them with one thing in mind only: is it blog worthy? That it may not be, that it's a very peculiar way of looking at the literature, and I find these things, they just pop up to me. So
yeah, Michele, they just pop up in his blog automatically, and he just brings them here. Yeah, I like it. We'll see what people say, but I think it's great. It's a good idea.
Okay.
Thank you.
Okay.
All right, guys. Thank you.
Thank you.
Take care.
Much.
Bye.